We are presenting the risk of feeding a raw food diet, a systematic review and meta-analysis. For the presentation outline, we'll go over course connection, project summary, which includes research question, introduction and methods, then impact and results, reflection and acknowledgements. The course titled Biological Research Methods and Experimental Design served to provide a guided research opportunity to undergraduate students in biology. These are the components and activities we completed in class. About 50% was a team systematic literature review project, which is what we are presenting here today. About 40% was data workshop, which is different statistical methods and applying it to public data. And next, 10%, which is a final data analysis exam.
The purpose of our project was to conduct a systematic review and meta-analysis to answer the following research question. Does feeding companion animals a raw meat-based diet result in a higher likelihood of contracting zoonotic pathogens found in the GI tract?
RMBDs, known as raw meat-based diets, are gaining popularity amongst pet owners for their health benefits such as increasing the diversity of the animal's GI microbiome. It is also considered a more natural approach to feeding. However, a risk factor of feeding RMBDs is the potential of ingesting zoonotic pathogens such as E. coli, Salmonella, and for cats specifically, Toxoplasma gondii. For our methods, the database Web of Science was used to collect our studies. We included Boolean language into our search process to ensure that our studies assessed the intestinal parasites and pathogens, RMBDs, dogs, and cats. Studies were examined based on our inclusion-exclusion criteria and discarded as needed. Here is an overview of our study search process. Our study search process included identification, a screening period, eligibility, and the inclusion criteria of the articles in our quantitative analysis.
For the analysis, we used 3 different types of methods to compare within our 11 studies we used. So for the first one, we have % positive of control or treatment, and for reference, the control is pets fed commercial diets, and treatment was pets fed the raw food diet. So that equals the number of positive results for control or treatment divided by the total number of control or treatment times 100. Then next was the % of absolute difference, which equals % positive for treatment minus % positive for control. And lastly, the % positive proportion difference, which equals % positive for treatment divided by % for control. So to compare between our 11 studies, we used the % absolute difference. So here we have the zoonotic pathogens that we assessed in our study, which was E. coli, Toxoplasma gondii, and Salmonella. On the y-axis, we have our studies, and then on the x-axis, we have our % positive of pets that were fed RMBDs. So we can see trends for A through E and G through J. We see high % positives, with the exception for F, I, and J, but nothing statistically significant telling us otherwise. For our conclusion, we looked at the % averages, and our studies saw mostly that E. coli and Salmonella were found in dogs, and Toxoplasma gondii was found in cats. So for E. coli, dogs fed RMBDs were 24% more likely. For Salmonella, dogs fed RMBD were 12% more likely, and Toxoplasma gondii, cats fed RMBDs were 18% more likely. After completing our meta-analysis, we found that feeding companion animals raw meat-based diets were associated with higher rates of foodborne pathogens.
This project impacts the community because it provides pet owners with comprehensive information to make educated decisions about the care of their pets. Additionally, it generates awareness of the risks that can accompany these diets, not only to pets but also to humans exposed to the pathogens. After completing our meta-analysis, we learned that feeding companion animals raw meat-based diets was associated with higher rates of foodborne pathogens. We also found it to be challenging to find scientific studies that provided data, as under a dozen controlled assessments have been performed and only on a few common pathogens.
We would like to thank Dr. Mason for helping us closely with this research. We would also like to thank the UCF Biology Department for providing this course and the High Impact Showcase for students to be able to present their research. Here are our references. And we just want to say thank you for listening.